So now that we've talked about the discovery of DNA as the genetic material, now we'll take a little bit of time to discuss the structure and function of DNA. So what you see here on your screen is a piece of DNA, which is called the nucleotide. So the individual building blocks of DNA are called nucleotides. And so this little color-coded structure here is the nucleotide. And what we have is a phosphate group. So this is considered a polyatomic ion. It's a phosphate that's been bound to 3 oxygens and a hydroxide over here, and it's carrying this negative charge. So this is a phosphate group. And then here in the middle, we have our sugar, which is a deoxyribose sugar. It is a 5-carbon sugar, and we get a closer look at the structure of the sugar over here. So each one of these little points here represents a carbon, and then we've got this other carbon over here. And then we've got some hydrogens and some other things, and don't worry about these numbers here. We're not really going to discuss those very much. And then you've got this nitrogenous base. And so we call it a nitrogenous base because it's got all of these nitrogens associated with it. And so the base is going to give the identity to the nucleotide because there are 4 different bases. So this right here, this is what we refer to as a double ring structure because we have 2 rings that are bound together. So that's a double ring structure. And so what we'll see is we have 2 bases that have double rings, and those are called purines. And those are adenine, which we abbreviate with the A, and guanine that we abbreviate with the G. Okay, and you can see their structures are pretty similar. They've both got that double ring, just got this little nitrogen group in a different place. And then we have 2 other bases that have a single ring structure, and they have 6 carbons, so it's a 6-carbon ring, and these are called pyrimidines. And again, you can see that their structure is pretty similar. This is cytosine, and then thymine over here, which is in DNA. So we have adenine and guanine, and cytosine and thymine. And those are going to be our nitrogenous bases. Now, this up here with our sugar and our phosphate forms what we call the sugar phosphate backbone. And then the bases are going to kind of stick out in the middle of the structure, okay? And so here's what we've got. So here's our sugar, here's our phosphate group, and our phosphate group is going to be attached to this carbon, okay, what we call the 5' carbon, on each linkage. So the oxygen here is going to attach here, and then it's going to attach to the next carbon. And then our bases are going to attach to the 1' carbon, and they're going to kind of be in the middle over here, okay? And so we call this part right here our sugar phosphate backbone. Okay, so now we know DNA is the genetic material of cells. We know these nucleotides. We know what they're composed of. So now we're trying to discover what is the actual structure of DNA. Okay, so in the 1950s, early 1950s, we had a biochemist by the name of Erwin Chargaff. And what he did is he looked at the DNA content in different species and discovered that the adenine, thymine, guanine, and cytosine were not found in equal quantities. You know, before it was like, well, you know, they're going to be equal. We're going to have, you know, 25% are going to be adenine, 25% are going to be thymine, and so on. And that amount varied from species to species, but not between individuals of the same species. And in his work, he found that the amount of adenine was always equal to or very close to the amount of thymine, and the amount of cytosine was very close to equaling the amount of guanine, or A equal to T and G equals to C. And this became known as Chargaff's rules. So if we look back at our structure here, what this information told us is that adenine and thymine would pair. So we would have a purine and a pyrimidine that would pair with A and T. And then our G and our C would pair up together, so another purine-pyrimidine pair, because we didn't understand exactly how they paired up. They thought, well, the purines go together, the pyrimidines go together, but Chargaff's information told us that adenine always pairs with thymine and guanine always pairs with cytosine. So if you have 50% of your bases as adenine, then the other 50% have to be thymine. They come in equal percentages. Okay, so that's Chargaff's rule. So that gave us kind of our first piece of the puzzle. And then we had Linus Pauling, who was an American scientist. He was a structural chemist. He was trying to figure out the structure of DNA. And in fact, at the time, everybody figured he was going to win. He was going to be the first to figure it out. And so he started using this special kind of X-ray called X-ray diffraction. And based on his work, he proposed this triple strand model and couldn't ever get it to mimic what he saw in his X-ray photographs. And at the same time, over in Britain, we had Rosalind Franklin and her graduate student were also using X-ray diffraction. And what she came up with is this double helix. She was able to observe this kind of X pattern and figured out that there was a double helix, showed its helical nature. Now, we had another group, Watson and Crick. So Watson was an American, Crick was British, and Watson was in Britain working with Crick on the discovery of this structure. And they took Chargaff's rules, so they used Chargaff's rules. And then they had Franklin's photograph, which her colleague, Maurice Wilkins, had taken and shown to Watson and Crick. Franklin was not ready to release this information. She had this image. She wasn't 100% sure exactly what it meant, and she wanted to study it a little bit more. She was very thorough. But Wilkins took it and showed it to Watson and Crick, and they figured out the last piece of the puzzle in how to build this helical structure. So they published their model in the journal Nature in April of 1953. In this same issue, there were papers from Wilkins and his colleagues, and also Franklin and her graduate student Gosling, each describing the different aspects. And so in 1962, Watson, Crick, and Wilkins won the Nobel Prize for their discovery of the structure of DNA. However, Franklin did not get to win the Nobel Prize because she had died, and the Nobel Prize is not awarded posthumously. And when Watson, Crick, and Wilkins won the Nobel Prize, they really didn't give any credit to Franklin at all. But it was her picture that really put the last piece of the puzzle into place and allowed Watson and Crick to build their three-dimensional structure. And so this is their three-dimensional structure that shows their bases and then their phosphate groups and sugars here on the outside, and then their bases in the middle, and how it's going to have this helical nature because of the way that the bases attach to one another, and because of the way the phosphates and the sugars attach to one another. There's a really good documentary called The Race for the Helix that has Jeff Goldblum as James Watson. And it's a really good, I say documentary. It's kind of, there's a little bit of fiction in it too, but it's a really good presentation of the competition that existed to try to figure out the structure of DNA. So if we look at this a little bit closer, so this is our double helix, our helical model of DNA, and if we kind of straighten that out, again, we've got our sugar phosphate backbone here. Okay, and they are what we refer to as anti-parallel. They run in opposite directions, and that's why you see this 3' and 5' here. So their strands are running in opposite directions. And then in the middle, kind of making up what we would refer to as the rungs of the ladder, we have the A and T that are always paired together, and the G and C that are paired together. And here's a little bit more detail showing that 5' end and 3' end. And really, it's just the orientation of the phosphates. So on the 5' to 3' end, we kind of start with the phosphates here running down into the sugar. And then here on the 3' end, we have the sugar, and then on the 5' end having the phosphate. And by aligning it this way, we're able to kind of set our bases together. And then linking our bases kind of in the middle, making up the rungs of our ladder, we have hydrogen bonds. So these little dashed lines that you see here are hydrogen bonds. And we have 3 hydrogen bonds in between guanine and cytosine, and 2 hydrogen bonds in between thymine and adenine. So we know the structure of DNA, and we know that the function of DNA is to carry the genetic material. Okay, and now we switch gears a little bit and look at RNA. So RNA is ribonucleic acid, and RNA has a pretty similar structure to DNA with a few main differences. Number one is the sugar. In DNA, we have the sugar deoxyribose, and in RNA, we have the sugar ribose. Pretty similar. They're both 5-carbon sugars. The main difference is that deoxyribose has an OH and an H, and ribose has 2 OH groups. The other big difference is in DNA we have thymine, okay, but in RNA we do not have thymine. Instead of thymine we have uracil. Again, pretty similar structure. The main difference is this little piece right here. We don't have that piece hanging off in uracil. Okay. The other difference is that DNA is single-stranded, okay, and RNA is single-stranded. They still have the sugar phosphate backbone. We still have the nitrogenous bases in the middle, but RNA is going to exist typically as a single-stranded molecule. Now, it can fold upon itself. And if it does that, the complementary bases are still going to pair. Cytosine and guanine are going to pair, and since there is no thymine, adenine is going to pair up with uracil, and so they can form bonds there. So it can kind of create this kind of crumpled 3-dimensional structure by folding back on itself. Now, DNA stores the material. But in order for cells to actually be able to access that information, RNA has to be created, and that RNA is going to be used to direct the process of protein synthesis. So it goes back to Beadle and Tatum's idea of one gene, one protein, in that the DNA found within our genes, found in our chromosomes, pieces of that DNA are called genes, and each one of those codes for a particular protein. And RNA is what allows us to take that gene, take that DNA, and make the proteins. There are 3 types of RNA that can be made: messenger RNA, ribosomal RNA, and transfer RNA. And we abbreviate those with mRNA, rRNA, and tRNA, as you see there. So the intermediary, the first one really that was discovered was the mRNA, or the messenger RNA, that the information in DNA is transmitted to the ribosome using mRNA. And remember that the ribosome is the site of protein synthesis. Now, in a later video, we're going to take an in-depth look at the process of protein synthesis, which involves translation and transcription.